slot, actually, we have two speakers, right? We have Paul Humphrey and Christof Mitterer. Paul is a regional market manager for Thermo UK. And Christof? Okay. Is product manager at Thermo. And I guess he will at least mention or talk about micro pack columns. And of course, thank you very much to Thermo Scientific for the support of this meeting.
Thank you. Excellent. I hope you can hear me. I hope the audience elsewhere can hear me and see the slides. If not, scream. Yep. Just one thing first, Andrea. I arrived. Christof, unfortunately, is not here today. He's not available. He had COVID last week. He sends his apologies. Feeling better, but he's not appropriate to come here and sit in a room with all of you. So I will Christof's slides as well. In fact, all of these slides I'm presenting on behalf of colleagues, particularly Daniel Lopez Ferrer, who many of you will know. He works in our San Jose facility, Van Ling over here in Europe. And I'd like to thank them for their slides. If you like anything in the presentation, it's thanks to them and the work they have done and the slides they have given me. If there's anything you don't like in the presentation, it's me. It's my fault. I'm here. I'm the one who's here. Yeah, I'm the European Regional Marketing Manager for Life Science Mass Spectrometry and Omics and Clinical. So that keeps me quite busy. Great pleasure to be here. It's many, many years. I can't remember. I was trying to work out how many years it is since I've been to an Italian mass spec meeting, and it's too many. Yeah, it's and I think you guys have put together a tremendous program. The speakers you have on this important topic, I think it's a great program. So I'm looking forward to. I don't mean me. I mean the other speakers. So let's begin. What I want to talk to really is. Start, why are we doing single cell? Some of the other speakers have already alluded to this. I think all the speakers will have a view of it. I'll just give you, and obviously my view is slightly different because we're an instrument manufacturer. So, we have a slightly to one side view of things. I want particularly to talk about the challenges of single cell and how we, as a manufacturer, are trying to address those challenges and help the researchers, help you. Because single cell proteomics is not a thing for us, it's a thing for you as researchers. What we are doing is we are tool manufacturers. We try to give you the best so that you can do the research which is changing and improving the world for all of us. So I want to speak a little bit about those. I will talk through our workflow and the sort of things we are offering, but from a point of view of how I think they are the things you need to consider when you're looking at solutions for your research. I'm not going to give you a huge advert for buy this, buy that. To do that, that's not appropriate. I want to tell you the sort of things that we are thinking about and why we think they're important. And I also want to stress that I think particularly for single cell, it feels like a very collaborative thing. We are working with researchers, with other manufacturers, and everybody seems to be working together to solve this problem and to come up with advances and developments. And I think that's really good. There will be some applications scattered throughout, but I'm leaving a lot of the real applications for the academic presenters. But I also want to talk about our commitment to the future, where we see this is going, and why we think this is important and why we are involved. So, start with, why are we doing single cell proteomics? Why are you doing single cell proteomics? Why are you here today? The previous speaker said, and I wrote it down, heterogeneity is masked by bulk analysis. No, that's a nice statement. It really sums it up. If you do bulk analysis, you think, well, you get everything. And that's true to some extent. You get a huge amount of data. but you don't always get a lot of information because it's masked by the sheer volume of data. The way I look at it very simplistically is if you want to analyze a tree and understand the tree, you don't try and analyze the entire forest, because what you then get is information on the tree and lots of other trees, different types of trees and bushes and grass and birds and squirrels, other things, and it just masks what you're trying to look at. So you focus in order to understand what you want to do. And if you do the same thing for an organism, if you try and analyze the whole organism, you'll get a lot of data, but to get at the key information is more difficult. So you focus in. But even then, if you focus in, you look at perhaps the tumor. You get some cells are active, some cells are not active. So you need to go in even deeper focus. The closer you go in, the better your view is, the better resolution of information is what we're calling it, that you're getting. And I think that's very key. And once you get down to the single cell, you really get begin to get some understanding of the cellular heterogeneity. And this really shows up as a continuum of information. I'm not saying we don't do bulk analysis. Bulk analysis does give you some information, and that can be useful. You can identify biomarkers by looking at the bulk. You can understand protein expression, DNA, RNA expression levels. But down, increase your focus to the single cell level, then you begin to understand cell heterogeneity much better. You can compare the gene expression cell and develop a better understanding of the mechanisms involved, what's important, what's not important. The level beyond that, and I'm not going to go into that today, but it's to be aware of and to consider, is going for spatially resolved analysis. When you do that, you're getting also the where. You're understanding how the location in the tissue is important in the biological activity, and you can potentially resolve cell-to-cell interactions. That's an interesting topic perhaps for a future presentation. So there's a huge interest in single cell. We have a big audience here. We have more people online. You're all interested in this, and that's great. There is a lot of literature coming out, but as Erwin said in his talk, it's single cell analysis is very much in the early days, but it is already showing the ability to solve real-world biological problems. And you can see just from this selection of titles of peer-reviewed, some of the important areas that are being addressed by this type of research. Papers about COVID-19, major topics potentially affect every single one of us, not just in the room, but on this planet. So it is very, very important work that you are all doing. And the number of those papers is beginning to ramp up, and it's beginning to ramp up thanks in part to some of the people in this room and online who are, beginning to do this work, beginning to publish. And I think when we see, the graph extended next year and beyond, it will very much increase rapidly. It will begin to grow exponentially as work in this develops. So it's an important work to be doing. This is just a quick review of publications which have either single cell proteomics contained in them, and the numbers are growing. So what are the challenges? What are the difficulties in doing this actual work? There are several that you might expect, some perhaps you wouldn't expect, but there are several, and they are real challenges. There are things considered need to be looked at. Obviously, the first thing you need to do is you need to separate the cells and sort the cells. And you need to do that in a way which gives you a sufficient quantity of viable for analysis. So it needs to be done with a high quality. Once you have the cells sorted out, you need to prepare them. And there's lots of different mechanisms, lots of things you can do, but that can be difficult in itself. You have reduced the number of cells you're working with, so you have smaller samples, so you need to minimize sample loss. You need to be able to handle low amounts. If you single cell level, that becomes very, very difficult. Once you're looking at a small number, then you need to be able to do multiple repeat analysis in order to get sufficient data to be statistically representative. In order to do that, you need to be able to do either rapid analysis or high throughput analysis, ideally both. So sample throughput becomes a very important thing. That leads on to not just fast mass spectrometers, fast gradients, but also, sample automation, sample preparation automation. And I will talk about some of the things that have been dealt with then. The other feature that comes out of small sample sets is you need a very high sensitivity. You're looking at very small amounts of material, and the cells will have very small amounts of protein in them. And as it says here, not all cells are equal. A small cell, a blood cell, will have much less protein in than an oocyte. So you have very small levels you need to deal with. You need very sensitive analysis to be able to do that. And fortunately, mass spectrometry at least is an inherently extremely sensitive technology, and it is a rapidly advancing technology. Speed and sensitivity are two of the things which are increasing with mass spectrometry development over the years. I was at a conference a few years ago, and it was a 25th anniversary conference. So a colleague of mine did an analysis on how mass spectrometry had in terms of the speed and sensitivity of analysis over the previous 25 years. And it was interesting, the rapid change, it was unbelievable. And the way he put it across was, if car manufacturers had increased their technology in the same 25-year period, you would now be able to buy a car which traveled at about half the speed of light. So, it's orders of magnitude change in a relatively short period of time. And then the final step, and potentially the most difficult step, is you need to be able to quantify, and you need to be able to quantify precisely, because it's not just enough to know what's there. You have to know how much is there and how much the levels are changing, and to do that accurately and precisely. Biology is quantitative, so that's another thing to consider. So let's look at an experiment that some of you may be doing today, you may be doing in your laboratories tomorrow. This is just a simple view. If we take 5 different sample sets, they may be different time courses, different drug loadings. In order to understand them and analyze them, you need to not just run one, you maybe need to run 1,000 different sets from each one. So that's 5,000 analyses you have to do, and complex workflow to go through. You begin with your cells, you need to separate them. You need to do that by using a cell sorting, perhaps fluorescence activated system is the way. Often that has to be outsourced to a FACS facility. That means more time, more difficulty, potentially more expense. You then get the cells to a 384 well plate. Then you can begin the sample prep. Again, that's time consuming, potentially in losses and certainly delays. Then once you have the prepared samples, prepared perhaps by labeling, by digesting, whatever you want to do, then you're ready to take them for LC-MS analysis. And then once you've done that, the real fun starts, because then you hopefully have a huge amount of data to analyze, and then you need some very, very powerful data analysis software to pull out the information you really need from all the results you get. But just looking at the times for this sort of, if we do this analysis of these 5,000 samples, doing it label free, even with a 15-minute gradient, it could take you 53 days to do the analysis. It's a huge amount of time. And 15-minute gradient is reasonably quickly. 30-minute gradient, you look at 5 days. If you're doing an hour gradient, you could be looking at 7 months to do the analysis, label free. So you need to introduce some multiplexing to give you a chance of being able to handle this. Even if you just do 6-plex, your 7 month comes down to 3 weeks. Still a long time, but it's improving. If you do, which is more typical these days, 16-plex, 18-plex, then you're getting down to a few days, which is much more appropriate and much more capable of giving you a reasonable hope of solving some real world problems. And to sort of illustrate that, have a quote from the previous speaker, Erwin Schuf. Thanks to Orbitrap MS and TMT Pro technology, we can in principle multiplex 18 single cells at the same time. This enables us the possibility of running multiple 10,000 cell projects per instrument per year. And that is the sort of thing you will need to be able to do in order to get meaningful data and impactful biological insight. So it's an important thing to consider sample throughput. Another way of improving the sample throughput is to automate this middle section here, the cells, the sample prep. If they can be combined into a single step, then you have other potential for savings. And this is something that we have been very interested in developing, and we found that the best and easiest way to develop that was to enter a co-marketing agreement with somebody who's already solved the problem. So we entered a co-marketing agreement in the last year with Selenium, and that introduction of their workflow into our workflow is giving great potential for savings. And there is again a quotation from Meshler, who is sitting right in front of me. I am not going to read out what he says, because one thing I know about Carl is he speaks for himself, and I'm sure he will comment on later on. But you know, I think this is a very exciting development, and this is a great instrument which is looking to improve the throughput single cell proteomics. I don't want to go into details for two very obvious reasons. One, I am not an expert on this product because it's not my product. And two, the next speaker is an expert on this product. And so he is going to tell you everything you need to know, I'm sure. So I'm going to leave it to David in his talk. But very simplistically, it's an image-based system. You can see the lens here on the screen there, which lets you focus in and select the cells. They are then put into these small plates with the 16 wells, multiple 16 well sets. And within that, they then go through the whole process of sorting, of digestion, labeling, whatever you want to do, before they are then sent into the mass spectrometer. And this is a workflow we're experimenting with in San Jose, and we are very, very excited by the results we are getting. We're beginning to publish that. We've seen publications coming through the literature, which shows that this is a great technology, and I think it also has great potential for the future. I'm told that there is a possibility we will have 1 million nanowells in a single chip in the future. I hope I'm not saying anything wrong there. We will find out. But this is something that we should really look forward to if we want to do high throughput single cell proteomics. There are other solutions. That's just one. Something we developed and launched this year is a commercial pipetting robot, modified, optimized for nanoliter liquid handling. This is a product that we launched at the ASMS in June this year called the Accelerome. We sell the system, the robotic system. We also sell the sample kits to help you do the analysis. The beauty of this is it's totally integrated with the rest of the workflow. So the results from the Accelerome are passed into the mass spectrometers. The sequence list is handed over to make a very seamless analysis. I'll come back to the mass spectrometers in a little while, obviously. And then the final stage is passed to the data handling. We use Proteome Discoverer as one of our main tools. That's been upgraded this year to have a version 3.0, which features the chimeric system from MSAID, which is an artificial system which gives much better data handling, is able to pull out much more data. So again, very, very important to be able to do all of those things. Another quotation for you, this one from Ryan Kelly at Brigham Young University. To achieve successful single-cell proteomics, each stage of the workflow needs to be optimized, needs to be looked at, carefully considered. That's not just the mass spectrometry. You need to look at not just the MS, but also the LC, not just the LC, but also the chromatography columns. Everything needs to be optimized. And by making incremental changes at each stage, you can improve your workflows, you can improve your throughput, you can improve your sensitivity and your reproducibility. And I would urge you as researchers to consider very carefully all the steps along the way to make sure you get the best results. It's very important to you. Obviously, we have our own view of it. This is a recently launched LC system, the Vanquish Neo, which is optimized for nanoflow. It was launched, I think, September last year. So it's just over a year old, dedicated really for proteomics analysis. We've also just this year launched some new columns, which are very dedicated for this type of analysis. And these columns are very interesting. So I just want to talk to you a little bit about the column technology. Now, you know, if you've been doing proteomics using LC for a while, C18 is the most widely used chemistry. And over the last few years, that has gone through various improvements. The particles have become more regular. They tend to be spherical, so you get better stacking of them. They've got smaller in size, porous layers, typically using sub-2 micron particles in a great many of the column chemistries involved, but they are not perfect. There are still problems due to the size distribution not being perfect, which leads to increased back pressures, and that causes loss of chromatographic resolution, which is a failure in sensitivity, a failure in reproducibility, a failure in the, or not a failure, a reduction in the lifetime of the column. Now that's not a problem for us because we just sell more columns, but it's a problem for you. So we don't want to give you problems. That's not appropriate. But there is now the option to use a different type of technology with micropillar columns. And this has, we think, really changed the way that these column packing is looked at. It's a fairly novel approach. It reduces the back pressures by giving perfectly ordered and precisely controlled column diameters, diameters and the spacings, which gives improvements in resolution, reproducibility, sensitivity, and robustness, and column lifetime. It's true also. So you win on all levels. This little schematic, I like this because I like simple things to explain things to me. It just shows that if you have on the bottom a more ordered, then as you see as the sample goes through, there is much less fronting, much less tailing. There is not the spreading out, the diffusion of the chromatographic resolution. So very simplistically, there is easy to understand an improvement in the chromatographic resolution. And that's just a little simple visual aid to do that. How these columns are made is actually, I find personally, very interesting. They're based on silicon wafers. The actual microscopic structure is created using a photolithography system. Much has been done for many years for integrated circuitry and silicon wafers, obviously silicon chips. It gives a very, very precisely controlled, very highly reproducible structure. And you can see in this middle photograph a range of perfectly shaped, perfectly sized, perfectly ordered freestanding pillars, which are replacing, if you like, the spherical beads. But they are held in place because it is a single piece. It's a single piece of wafer, very easy to handle, very easy to deal with. And the columns, porous layers, it's bonded to C18, so you're getting the whole normal chromatographic procedure you have with typical C18 chemistries, but in a much more controlled way. What they actually look like, people say, well, yeah, how does that actually look? And this photograph on the left shows it's very just The wafer is just held in an aluminium holder. It has the normal capillaries front and back. They are, amenable to Viper fitting, so it's very easy to just adapt it and to, fit it into your normal system. You have different loadings available. We have a specific low load version, which is, we think, ideally optimized for single cell proteomics, and we'd recommend that. But there are other versions available, and more will be coming available over the years as we begin to extend that. The reproducibility is very good indeed. This shows 9 peaks on 10 columns, and you can see in terms of both retention time and peak width, very, very reproducible data. The percent CV for the retention time, less than 1%, so very, very reproducible, which really proves what I said about the control and reproducibility of the structure is reflected in the reproducibility of the chromatography and the data coming out. And when we look at real data, at peptide protein identification, we can see in this is a direct one-on-one comparison with a pull tip column, and in every sample loading, every gradient microarray is better. There is an improvement at every stage. So, it's something to certainly consider and something we're very excited about as another step along the road to single cell proteomics. And this is just some data, not from us. This is from Erwin Schuf, who was speaking earlier on. This is very early data that he got using the 50 centimeter low load column. And again, different loadings, different sample gradients, but improvements, whether you're looking at PSMs, peptides, or proteins, all across the board, there are improvements. The next stage, we've done the LC, we've had the column. Before we go straight into, we can either go straight into the mass spectrometer. We can also look at another intervening stage. This is one option that is available, and this is the FAME system. This is a field asymmetric ion mobility system, which gives another orthogonal level of separation before your samples go into the mass spectrometer. And you can see from this example, after we launched the FAMES, we began to investigate it for a range of different samples, not just proteomics, but we now have a version which is optimized for proteomic operation, and looking at protein groups, the number of protein groups you see with and without FAMES, you do get an improvement. Not in all cases, but you can optimize it and tweak it. And again, it's another step along the road, another improvement. And if you make incremental improvements at all stages, then your overall workflow does improve. And again, there begins to be literature coming out which illustrates that. You don't have to believe me. Search the literature, the results are coming through. So, mass spectrometers. This is always my favorite bit. This is a Mass Spectrometry Society meeting, so I can't sit here and not talk to you about the Orbitrap. It's personally something I'm very passionate about. I've been very lucky. I've worked for this company for many years. I have known Professor Makarov for 30 years, nearly. And I will tell you a secret, as long as you don't tell anyone else. Sure. Yeah. Thank you, Andrea. I was fortunate enough to be there in the room the day Alexander got his first results on his first Orbitrap. I saw the first spectrum and I was very excited. I was one of the first 2 people in the world to see this, and I was one of the first 2 people in the world to tell him he'd made a mistake. The reason I said that, and this is why this is an embarrassing tale, I didn't believe the data. My managing director did, because it was too good. We said, this can't be right, Alexander. You must have made a mistake. Fortunately for me, for you, for all of us, for mass spectrometry, he didn't listen. He repeated it. He got better results the second time and even better results the third time. And the rest is history. Orbitrap mass spectrometry has, and this is not marketing hyperbole, it has changed mass spectrometry. There are only really fundamentally five types of mass spectrometer. Orbitrap is one of them, and it is the most remarkable. And you don't have to believe me, just look at the literature. This is a view of the number of publications that have been coming out since we launched first commercial Orbitrap in 2005. The number of publications is growing and growing and growing. In the last 12 years, over 75,000 peer-reviewed articles have been published on Orbitrap technology. In 2020, 1.5 articles per hour were being published using Orbitrap technology. If you just look at Science and Nature, the number of publications on Orbitrap is not only more than every other mass spectrometer type, it's more than every other mass spectrometer type added together. So Orbitrap is a tremendous advancement. It's a wonderful mass spectrometer. And this is true also in single-cell proteomics. If you look at the peer-reviewed papers for single-cell proteomics, they are absolutely dominated by Orbitraps. And if you also published presentations at a conference, this is from ASMS last year, again, it's dominated by Orbitraps. And the interesting thing is, it's not just the brand new latest Orbitrap that's dominating. These show many older Orbitraps still working, still producing excellent data. So, this is obviously a technology that we hold very dear to our hearts. I personally have been very invested in it and very pleased to be able to watch the development of Orbitrap and see how remarkable it is. That's not to say there aren't other mass spectrometry technologies out there, of course there are. But Orbitrap is well up there and needs to be, I think, recognized as such. And this just shows another example of that. This is some work from Professor Slavov's group. I know Nikolai will be talking later in the afternoon. This shows some of the work he has done, and I think we'll be presenting on later. He's the development he had 2017. He's single-cell proteomics by mass spectrometry, scope MS. This is the methodology he came up with. Erwin also mentioned it in his talk earlier this morning. And Nikolai has evolved this, scope 2 in 2019, Plex DIA, P-scope this year has come out. But all his great advances have been done on an 11-year-old Orbitrap. So the technology is solid. And I don't want to, in this talk, this is a proteomics meeting. I don't want to go into how the mass spectrometer works. If you want to do that, talk to me, talk to my colleagues. We can go over that. I just want you to know that it is a tool that is available and provides, I think, important and key results. And that really is a nice slide to then look in to sort of segue into a timeline of the evolution of single-cell proteomics. It is a relatively new area, and as Erwin said, it is a rapidly growing, still in the early days. If we look before 2017, before Scope MS, then single-cell Western blots were available. It took time, and you were having maybe 10, 11 targets per cell. Mass cytometry came along. That gave you the ability to do more. The next stage was beginning to do single cell by high resolution accurate mass LC-MS on a large single cell like an oocyte. And that began to then give us the scope and the hope that we could actually improve the sample prep, improve the sample handling, improve the throughput. We already had the sensitivity by bringing in multiplex labeling using TMT Pro. We could do more and more samples. We could get the times down to something which was handleable. Then we move on to where we are today. Automated Selenium system we will be hearing about next. Again, giving more and more advantages and more improvements. Where do we go in the future? Again, see a quote this time from Nikolai. We need creative experimental designs and methods to make single-cell proteomics broadly accessible. You as researchers need that, you want that. We as tool manufacturers, whether we're making the mass spectrometers, the LCs, the columns, sample handling robots, whatever, we need to work with you to help produce better tools for you so you can do the research, so you can do good multiplexing, you can have short analyses, you can have high throughput, you can have high sensitivity. We are very happy to collaborate, to partner with you, to improve the technology, because this is something that is important to everyone. As Francesca said in her talk, you know, this has real biological application across a huge range of disease states. You know, we think that's very, very important, and we can see going forward, making single-cell proteomics more widely available is a very important process, and we are committed as a manufacturer to doing that. We're working on the technology improvements across not just the hardware, but also the software. We're collaborating with biologists, with technologists, with protein chemists, so on and so forth, and other manufacturers to understand these challenges I've outlined better and to look for novel ways of circumventing or bypassing these difficulties to really drive the science on and to change the world for all of us. This is the workflow we have at the moment, but it's just an example. There are other solutions, there are other things we can offer, there are other things other people can offer. You as research scientists doing this sort of research, tell us what you want, and collectively we will try and give you that, because you need these to work. And it's very much an ongoing roadmap. This is just the last couple of years, some of the developments, TMT moving to 16 and then 18, SureQuant coming in, the Vanquish Neo launched last September, Proteome Discoverer with artificial intelligence came out earlier this year, SLM1 integration, Reticle, I haven't even mentioned, Erwin made a brief reference of that, and there are more steps along the road. It's an ongoing journey, and we hope you will join us with it. And you know, we hope that we will together be able to take single-cell proteomics forward into a very exciting area where real biological improvements and discoveries are made to the benefit of everybody. So thank you very much for your time, and it's been a pleasure to be here today. Thank you.
Thank you very much for your talk, for being here today, and was very, very exciting, plenty of data. Just a note, I mean, this field is clearly led by Orbitrap technology. That's a fact, no question about that. Just to mention that we will also have speakers today showing what the TOF technology is doing in this field. Just to
absolutely no. You need to investigate everything, every option. Some options are better for some things. It's like the way I always say it is like you buy a car. If you want to just go to the shops, maybe you don't buy a Ferrari. You know, you have to buy the right tool for the job you're doing.
So, any question from the audience? Guys, come up with question, otherwise number. Yeah, please, Simone, go ahead.
So actually, I have a science question related to you have such a broad overview of lots of people who work with the Orbitrap and they have the latest innovation, etc. My question is related to so the way we classify cell types today, it's basically based on how they behave and how they look at the microscope, right? Which with the technology we have today seems like medieval, right? Because I'm wondering if any of the people that you work with had thought about proteomics can become basically a better classifier, like we will rename cells based on the type of data that we are generating with single cells today?
Yeah, I mean, I think that is a very, very good question. And I'm not a cell biologist, but I think absolutely that will happen. Absolutely, yeah. But you're right, it is almost medieval, the way we do it. And the world is changing, and it's changing in a good way. So yeah, watch this space.
Awesome.
Happen with multi-technology and bacteria.
True, yeah.
That's right. That's it.
Okay,
Yep.
thank you very much again.